Hello all, and welcome to today's APS webinar entitled Using Enzymes to Digest Muscle and Cardiac Tissue Models, presented in partnership with Worthington Biochemical. My name is Jacob White of the American Physiological Society, and I will be serving as your moderator today. Now, in today's session, Dr. Thomas Molley and Alice Balayan will present their research on muscle regeneration and cardiac model systems and explain their research findings on isolation and characterization of human skeletal muscle stem cells and how these stem cell populations change with age. Additionally, they will highlight tips on how to best optimize dissociation protocols for a variety of musculoskeletal tissues and non-muscle tissues. Now, a few housekeeping notes. Respect for our speakers, we ask that you refrain from photo or video capture of the presentations in whole or in part. The session will be recorded and available for future viewing through a link on the registration website. Recording or taking photographs of another person's presentation without their explicit permission is prohibited. Now, at this time, all attendees, except for the speakers, are in listen-only mode with video off. As moderator, I will be happy to raise questions that are entered into the Q&A function located at the bottom of your screen at the end of the session. The session will end today at about 12 p.m. Eastern. Now, before we begin, I'd like to thank our partners and sponsors at Worthington Biochemical. And before we get to our introductions, I'd like to take this time to launch a brief poll to help guide today's session and our educational efforts. Please take a moment to respond. Thank you. Thank you very much for your responses. Now, to get to the introductions, it is my pleasure to introduce our speakers. First, Alice Balayan is an MD PhD student at the University of California, San Diego, where she performs doctoral research on the age-related decline in skeletal muscle regeneration. Tom Molley, PhD, is a Ruth Kirsten postdoctoral fellow in the Department of Bioengineering at the University of California, San Diego. He earned a PhD in materials science and engineering from the University of New South Wales, Australia, where he developed advanced biofabrication strategies for tissue modeling. Now, on to our program. Tom, please begin whenever you're ready.
All right, will do. Thank you for that wonderful introduction, Jacob.
Great.
All right, so hopefully everyone can see our screen just fine. And so our research with Alice and mine in our laboratory is highly driven by the fact that age causes a lot of decline, particularly in cardiac and muscle function. So most adults have some sort of form of cardiovascular disease, and it's a leading killer of adults worldwide. And this is particularly, a particular pathology that seems to appear more as people begin to age and get older. A lot of patients at young ages aren't really afflicted by these until they begin to reach an older age. In addition, muscle mass at older ages is highly correlated with all-cause mortality. In particular, there is an appreciable notice in the decline of muscle regeneration. So both of these kind of muscular tissues seem to have a large effect with age as we progress. And one thing that's also very similar with both these tissues that they're highly dynamic. As most of you guys are aware, our hearts and our muscles are constantly contracting and pumping throughout our entire lifespan. And in particular, what's also noticeable about this is that these cells most of the time do not proliferate. So we have proliferative cells in the muscle that build the muscle, but these muscle fibers themselves, in particular cardiomyocytes, do not continue to divide and keep replacing themselves as we age. So these individual cells can be decades old by the time that we're an adult. And so this can be a key driver of some of these issues as we age. And so this sort of begs the question, well, we have this really complex contractile dynamic tissue sets with these unique cells that don't really proliferate much as we get older. If we want to understand these cells more, it kind of begs, well, how do we actually model these in the laboratory? Well, the main way that most people do these is just using established cell lines. And this is really challenging for, say, cardiomyocytes, particularly myofibroblast cells, again, because they don't continually reproduce and produce themselves. And so what a lot of people do, which is a very strong method, is using patient-derived cells. And these can also come from animal models. So you take actual tissue from particular patients or models, digest them down to pull out the individual cells that we want, and then use those for downstream assays. Another method that people begin to use, particularly in the cardiac space, not as much in the musculoskeletal space, but moving more in that direction, is using organoids. And so these are derived from stem cells in which we can differentiate them into the particular tissues that we want. With that native heterogeneity, hopefully trying to recapture and recapitulate those patient-derived tissues, but in the laboratory dish in a more complex organotypic model. And then finally, people use actual animal models to do a lot of these studies. Although, we can't do a lot of these on actual human patients, right? And so what our lab focuses on is the two different aspects in the middle. So patient-derived cells, where we isolate those cells, and also using stem cell-derived organoids. But one particular challenge with doing both of these models is isolating those cells from those heterogeneous tissues. So as most of you guys can probably appreciate, the extracellular matrix that native tissue builds and that even organoids create themselves is full of a cacophony of different proteins and materials. And so if you do proteomics on, say, a muscle tissue or a lung tissue, you're going to find over 60 to 70 different types of matrix proteins. And what makes this complicated is that different cells like to reside on different protein materials. So say cardiomyocytes, contractile cells might sit more on a collagen 4 matrix, where, as say, endothelial cells or some satellite cells might sit more on a basement membrane-like matrix. And so when we're trying to pull these cells out of these tissues, we need to have different kinds of enzymes to digest these different kind of matrix proteins. And what's challenging is that enzymes digest at different speeds. And once these cells have been removed from their individual, preferred matrix that they typically bind to, they can begin to undergo apoptosis if we're not careful. And so again, if you try and digest a tissue for 48 hours or something to really remove everything, the cells probably aren't going to survive. So we're kind of on a clock here. And so this is sort of a focus that we've been trying to work around, and something that we want to present to you guys today, is to show us the kind of models and research we do, and then how this enzyme digestion, which is a really critical aspect because it controls the quality of the cells and number of cells that we get from our models, how this sort of plays a role within our research. And to do that, we're going to talk about two stories. One will be human skeletal muscle regeneration story, mostly work that Alice Balayan has been working on, as well as then cardiac engineering stuff that I have worked on. And so with that, I will pass it off to Alice here.
Hi, everyone. So I'll talk a little bit about my research in the human skeletal muscle regeneration field. So to start with, I'll talk about background. So skeletal muscle is the most abundant tissue in our body and is responsible for producing movement, sustaining body posture and position. And it also helps with stabilizing joints and major contributor to the body's metabolism. It is composed of highly organized and densely packed myofibers, blood vessels, nerves, and connective tissue. And within the skeletal muscle, there are the stem cell populations, like Tom mentioned earlier, the satellite cells and fibroadipogenic progenitors, that they play an integral role in muscle regeneration. So satellite cells themselves reside in the skeletal muscle, and they're a quiescent and dormant pool of stem cells in the healthy muscle, but following an injury or certain stimuli, they're able to activate, proliferate, and eventually differentiate into myofibers that can regenerate an injured area. Fibroadipogenic progenitors, on the other hand, play a supportive but a key role for satellite cells during their muscle regeneration. With that, they secrete certain factors that help with their proliferation and self-renewal, and they themselves are able to differentiate into either fibroblasts or adipocytes. Fibroblasts are able to modify the extracellular matrix of their microenvironment, and at appropriate levels, they're able to provide certain cues to direct proper regeneration. However, accumulation of too much fibroblasts can cause fibrosis, and similarly, differentiation of FAPs into too much adipocytes or too many adipocytes could cause fatty accumulation. With that, in normal regeneration, there's a delicate balance of satellite cells and fibroadipogenic progenitors and their function. However, with age, there's a decline. There has been seen decline in the quiescent cell population of both satellite cells and dysregulation of fibroadipogenic progenitors. There's secretory factors and predominant commitment of fibroadipogenic cells to fibroblasts that then increases stiffness of the muscle. And with this, all of these functional dysregulations then cause skeletal muscle regeneration decline with age. And as you could imagine, this could have implication in terms of mobility limitations with increasing age, with increased fall risk and functional limitation. And so I was really interested in studying some of these changes, and a lot of the literature that currently exists is based on mostly animal models or animal-derived cells. So then I was interested in looking at like some of these changes with human skeletal muscle. However, when I first started this project, I kind of encountered a big issue where the isolation of simultaneous isolation, specifically for both satellite cells and fibroadipogenic progenitors from human skeletal muscle, was understudied. So for me to be able to study these cells in culture, I had to pull them out of the native tissue. For that, I had to do some sort of dissociation. But when I looked into literature, the satellite cells and Fab co-isolation was, to say the least, difficult and poorly studied. The satellite cells themselves compose less than 5% of the myonuclei in the muscle. We get very limited size of biopsies from human patient samples. There's a lack of detailed protocols and systemic and comparative analysis for both or individually satellite cells and Fab isolations, and there seems to be a lack of consensus in the literature for which isolation methods to use. But so when I did my literature review, I found that a lot of these studies that have been published use either a combination of disbase or collagenase. And so for me, I was interested in isolating both at the same time. So I had to come up with a enzyme formulation that would allow for isolation of both of the cells at high yields at the same time. So when you look closely, you could see that a lot of studies that did fibroadipogenic progenitor isolation by themselves use some sort of collagenase 2 and collagenase 1 combination, mostly concentrating on collagenase 2. And for satellite cells, they used both the disbase and a collagenase concentration, enzymes. But a lot of these studies use very different enzyme concentrations and units and like varying times for dissociation. So I kind of had to formulate my own. And for that, my hypothesis was that the disbase collagenase cocktail will enable dual isolation of satellite cells and Fab, so it significantly increased years from the human muscle biopsies. And to test this hypothesis, I was able to recruit about 16 patients, 7 females and 9 males, with the respective ages here. From 2 different muscle groups, semitendinosus and gracilis. I was able to get these muscle samples from patients undergoing ACL reconstruction surgeries. So then I used these muscle samples to isolate the satellite cells, and here's the schematic of my isolation. So here's a picture of the muscle samples that I would get. First, I would remove any of the kind of contaminating muscle tissues in the muscle, such as fat, circled right here, or tendons. And then I would use scissors and forceps to finely mince the tissue into a fine slurry, as you could see over here, and eventually put these the muscle slurry and a digestion buffer with my disbase and collagenase formulation. And I tested two different formulations for the enzymes I mentioned before, and I could show the different formulations later. And so at this point, after the dissociation, I'm able to obtain a heterogeneous cell population, which I then start removing some of the more not contaminating, but the cell populations that I'm not necessarily interested in. So first, I would remove the red blood cells by lysing them. Then I would use fluorescence activated cell sorting by staining the cell populations with markers for one satellite cell, CD56, to identify satellite cells in culture, and then use CD45 for identifying immune or hematopoietic cells or endothelial cells with CD31. So then use these as exclusion markers. So then I'm able to sort out CD56 positive, 31 negative, and 45 negative population as my satellite cells, and the triple negative population, which would be enriched in fibroadipogenic progenitors and more fibroblast-like cells. So when I tested the two different digestion methods, and here are the units that I used. So both of the digestion methods had 2 units per ml of disbase, and they had a combination of collagenase 1 and 2. Digestion 1 had about 50 units per ml, which was when I looked into the Wharton recommended usage. This was the minimum recommended for each enzymes per ml of digestion buffer. So then I used a 60-minute digestion, indicating, so digestion 1 was a very gentle digestion versus digestion 2. I doubled the concentration of collagenase 1 and 2. And increased the time a little more. So with digestion 2, I was able to get a lot more cells. So total number of cells per gram of tissue was a lot higher in digestion 2. So then that also translated into having a lot more satellite cells, as indicated here by CD56 expression, and also fibroadipogenic progenitors. And also these yields were compared to some of the reported values in the literature, and you could see here the range in dotted lines. So then, since digestion 2 yielded more cells, I used this digestion method and characterized the cells that were pulled out using the digestion 2. So here we could see some of the characterizations I do in culture. So I was able to get pure satellite cells and Fabs and expand them in culture. First, I validated their lineage specific marker expression. Here, fibroadipogenic progenitors were positive for CD140A and also TE7, both markers for Fabs and fibroblast-like cells, since satellite cells expressed PAX7. I also validated that using flow cytometry to make sure that the surface markers are maintained in culture after expansion. Following, then I demonstrated that the satellite cells in culture are activated and proliferating, given their double expression of PAX7 and MyoD. And also then I demonstrated that about half of the satellite cells and also fibroadipogenic progenitors in culture are actively proliferating. And then I also demonstrated that both satellite cells and Fabs can be expanded in culture. But our satellite cells kind of reach senescence about passage 5 or 6, while Fabs could reach higher passages. So then I moved on to further characterization. Satellite cells, I was able to demonstrate in culture that satellite cells can fuse together and form myofibers after expansion in culture. And here you could see that satellite cells are able to form these multinucleated structures or myofibers that stain positive for myosin heavy chain. I was also able to demonstrate that fibroadipogenic progenitors can differentiate into fibroblasts after expansion. So after stimulating the fibroadipogenic progenitors with TGF-beta1, I was able to see upregulation of fibronectin, which is a marker, which I used as a marker for fibroblast differentiation. I was also then able to demonstrate that fibroadipogenic progenitors can differentiate into adipocytes after expansion, again, after adipogenic induction in culture for about 14 days. I was able to see robust upregulation of fatty acid binding protein 4, which I used as a marker for differentiation. Also, you could see here staining in green for fatty acid binding protein 4. So part one conclusion was that a digestion 2 method allowed me to isolate satellite cells and fibroadipogenic progenitors from the same patient samples. And these cells are able to expand in culture and maintain their phenotype and differentiation capacity. And then I was interested in further investigating some of the age-related changes that are happening in these patient-matched satellite cells and FAPs. So for part two, I'll look into some of the pools of the satellite cells and FAPs, and next look into their function across age. So for part two, then here. My hypothesis was that in older patients, a significant decline in the satellite cell proliferation and fusion capacity would be coupled with maintained FAP proliferation, and which would contribute to impaired skeletal muscle regeneration with age. So for that, then I recruited about 52 patients across 3 different muscle groups, gracilis and semitendinosus, and also gluteus max for this study, where I was again obtaining the gracilis and the semitendinosus muscle from ACL reconstruction surgeries, while glute max from hip replacement surgeries. So then I used these patient samples to look into some of these cellular changes or the cellular composition changes that were happening across age. So one of the most interesting result here in my data was that satellite cells across all 3 different muscle groups, we were seeing this drastic decline in the pool of satellite cells that I was able to quantify using flow cytometry. While fibroadipogenic progenitors, there was a tendency of a slight increase, I guess, in the pool. However, the trends were not significant. For endothelial cells, we were seeing trends of decreasing endothelial cell population in the muscle, potentially indicating less vascularization of the muscle as we're aging. But again, the trends were not significant. For immune cells or CD45 positive hematopoietic cells, again, we're seeing trends of increasing CD45 positive cells, and the only significant one was part of the gracilis group. And I think this was interesting to see, validating some of the data that has been generated in the literature, that the satellite cell pool is declining. But I think it was really interesting to see that the fibroadipogenic progenitor population was maintained in culture. So then to look into this data further, I also stratified my data based on sex, females versus males. And I identified that the cellular composition changes that we were seeing with age were not influenced by sex. So here I demonstrate with 2A ANOVA that a lot of the variation and variability in the data was due to age, not sex. Next, I looked into the functions of these cells across age. So I demonstrated here that there was a drastic decline of the proliferative satellite cells in culture, while first fibroadipogenic progenitors, it was somewhat maintained. And for differentiations, also I'm seeing a decline in the fusion capacity across age, while for fibroadipogenic progenitor, differentiation into fibroblast was maintained, but into adipocytes, it was declining. And one disclaimer here, obviously, my age range is anywhere from like 10 to 40 years old. So one of the things that I'm working on right now is to expand some of these differentiation to include more adult and older populations. So conclusion for second part was that I'm seeing all of these changes, including that satellite cell quiescent pool was declining with maintained fibroadipogenic progenitors, and the function of satellite cells indicated by fusion capacity was also declining in older patients, while fibroblasts maintained their FABs maintained their fibroblast differentiation. So some of the outlooks for me is to look into some of the transcriptional epigenetic changes that are happening in the satellite cells and Fab populations, and to understand why there's this decline of muscle regeneration as we're aging, kind of getting more into the molecular side of things. And then the hope is to then also look into the interaction between satellite cells and FAPs and how aging affects that kind of crosstalk, and try to find some therapeutic interventions where we can kind of remedy that decline in the crosstalk and also function between satellite cells and FAPs. And so with this kind of, I'm going to conclude my presentation. I'm going to pass it back to Tom.
Excellent. Thank you for that wonderful section, Alice. I'm always super excited to see all of that data. And so to change gears a little bit and jump into our cardiac engineering project, we're using organoids to do this. And one challenge with organoids is that they're not quite as robust as regular human tissues, right? And so before we can jump into the age-related challenges with cardiac engineering, as we can with skeletal muscle engineering using human tissues, we want to improve our organoid models first. And so when it comes to cardiac organoids, there's the good, there's the bad, and there's the ugly. So the good part of cardiac organoids is that they capture a lot of good cellular heterogeneity. They can enable a lot of good 3D cell-cell interactions that we can't get when we do 2D monolayer cultures or 2D differentiations into cardiomyocytes. And what's beautiful is that they contain native vasculature. So compared to most other organoid systems, you'll hear a lot of people talk about kidney organoids or brain organoids trying to put blood vessels in. The nice thing with cardiac organoids is they naturally produce them. The bad, though, is that most of these organoids, if not all, don't have any accessible hollow cores or channels that actually pump anything. You think of a real heart, it literally has fluid flow pumping through it, and that form and function really drive a lot of the maturation and proper heterogeneity and structure of the real organs. They're also quite difficult to properly characterize live, both their electrophysiology and contractility, right? These are 3D kind of balls. And this sort of ties into why it's really important for us to have good digestion protocols so that we can pull these cells out later and do some of these more robust characterization techniques. But the ugly is that we're completely missing that proper native cellular organization. So not only do we not have those hollow cores, but we don't have the cells perfectly aligning. We don't have them stratified in the correct orientation. There's a lot of intra-batch as well as batch-to-batch reproducibility. So you make one batch of organoids, within that batch, there's a lot of problems. And then you do another one a few weeks later, and again, they don't look identical to the original ones. And so to give you an idea of some of that structure here, this is a cardiac organoid that we've made on the left. You're looking at the actin structure, and you can see it's not really a well-organized system. We can compare this to, say, just an intestinal organoid, not even real intestine, and you can already see that some of these other systems have much more reproducible cellular organization, and that's usually because they have good stem cell progenitors that can produce the rest of that, model tissue. Whereas when we have, we're doing skeletal muscle engineering or cardiac engineering, there isn't really a great architect cell type that is directing the function and form of these tissues. And so if we take this a step further, so you can see the difference between our cardiac organoids versus other intestinal organoids, then if we look at real native structure of actual tissues in the body, and you can tell that we're nowhere close to the level of complexity of spatial organization that we find in the actual human tissue. Now, these ones aren't the heart, but I think they really demonstrate what kind of beautiful architectures that the real body can make. And we want to try and get closer to this so that we can actually use these models to accurately understand, how some of these pathologies and age-related problems might actually be affecting them. And so the main question that I've been trying to focus on is, well, what is the body doing differently, especially during embryo development, to give cues to let these cells create the proper architectures and structures as they differentiate? And how can we then give them those same cues and guide them towards that actual structure themselves? So my particular approach that I'm using during my postdoc is to use geometric confinement and compliant substrates for organoid generation. And so to do this geometric confinement, I use a cool technique called microcontact printing. So the way that this works is we take a glass cover slip and we put a hydrogel on top. In this case, polyacrylamide, 10 kilopascals. So, a wet polymer substrate that's mimicking the ECM. What's nice is that polyacrylamide naturally does not allow for cell adhesion. So what we can do is we can chemically functionalize it with hydrogen. And then we can take these PDMS molds. And what these do is they look like Lego bricks with raised surfaces on them of different shapes. We functionalize those with protein, then we turn them upside down and smush them into our hydrogel and peel it off. And what we're left with is it's still a flat 2D surface of hydrogel, but now we have these patterned islands of protein where the cells can bind to. And so not only can we now grow these different islands of cells, but my old PhD advisor, Christopher Killian, had shown back about 15 years ago that by controlling the geometry of individual stem cells, we can affect their differentiation outcome. So a lot of these cytoskeletal tensions and shape cues can actually drive actual functional outcomes from stem cells. And so the main question we wanted to ask was, can we do this with pluripotent stem cells and try and convert them into organoids at the same time? So can we see how geometry and these soft materials like polyacrylamide at different stiffnesses, can we use those in conjunction with morphological cues that we typically use for organoid generations and create something that's even better than what we currently find? And so we had to come up with a particularly unique protocol to do this. This was combining 2D protocols and 3D protocols for differentiation because we have this quasi 2D. So as you can sort of see here from this timeline, and again, this is one of the reasons why it's so challenging, is that these organoids form within about 7 to 9 days. So they do mature later than that, but after this stage, they don't proliferate too much. They stop growing. And so we'll have this monolayer at day 0, and as we give them these cues, they begin to grow as these domes until they form a giant ball at day 7 or 9, which then begins to contract. It can lift off our geometric patterns, and then we can have a functional organoid that we can then try and assess and do things with. So this is sort of just to demonstrate that we can make these arrays of these contractile organoids, and you can sort of see here on the left, we have a cardiac troponin T reporter line. So cardiac troponin T, a key protein in the contractile sarcomeres of these cardiomyocytes, fluorescing green where we have actual contractile cells. So we can make these large arrays of these organoids. We can then do immunofluorescence staining on the whole organoid, right? So this is just 2 different organoids that we had pulled up. Sort of showing alpha-actinin, which is again, a key contractile protein that you find in the sarcomeric structure in these organoids. And so we could sort of pull that apart and we can sort of look to see, yes, we are getting that intercalated disc, that sarcomeric structure that we're finding inside of these organoids, which is nice. So we're able to produce them. And again, but you can see here, look at how unorganized in terms of the direction that they're in. They're not perfectly aligned. There's gaps, there's holes. And we don't have like a perfect monolayer of, cardiac fibroblasts sort of in between with endocardial cells on one side and epicardial cells on the other side. So we're getting close, we're getting closer, but we're still not quite there. And so one other way that we try and characterize these is we use contractile algorithms just using bright field. So this is before we do digestions, when we're trying to get a rough idea of how the entire structure works together. We can take videos of these organoids as they beat, and then we can look to sort of see how often and how hard they contract and pull up parameters from that. So just as a little aside to show you, we had compared, say, embryoid body differentiations from pluripotent stem cells to our organoids derived from micropatterns, and we found from a lot of these different parameters from our initial test that we had done that were pretty similar in terms of actual contractile function, at least in a 3D context. But what was really interesting is then we figured, okay, well, we have these different, this tool to control geometry. What if we just change the size? Right, so we start with a circle of these different radial diameters, and you can sort of see here in this clear image set. So these are heat maps where we've taken confocal images of these organoids as they develop, and then merge them down and then average out over 20 to 30 different replicates of their immunofluorescence staining. Again, this is in 3D prior to digestion, and sort of looking at, okay, when we change the diameter of these initial cells, like how does this affect the organoid? And what we found is that when we have really small diameters, so about 250 microns or smaller, we have no structural organization of these cardiac cells. But as we get larger, we typically then sort of see a ring begin to form, where these contractile cardiomyocytes begin to appear on the outer edge of these patterns. And when we get super large, they sort of lose their capacity to differentiate. So it's clear that there is a massive geometry and geometric effect here. As we just double the size of something, we've completely changed the structure of how these cells are organizing themselves. What was also interesting is that CD31, as Alice mentioned, a vascular endothelial cell marker, kind of followed a little bit of the opposite trend. So in the smallest organoids, 250 micron diameters, we found that they typically were semi-evenly distributed, if not more on the edge sometimes. But as they got larger, we typically found more of these vascular cells on the inside. And this intuitively makes sense. You have a large structure, you're going to start to have hypoxia or a lack of nutrients in the middle of the organoid. And this part is likely to drive either endothelial cell differentiation or at least migration towards these more hypoxic regions. And to give you a little teaser as well, we began to look at how this is affected by, say, moving the middle of these patterns as well, these center colonies. And what we found very interestingly is that this had very minimal effect on those vascular cells. So if at day 1, we've removed the middle, by day 7, they've sort of closed that gap and they've still created these giant ball structures that look the same by eye. What we found is we have way less contractile cells on the edge while we have no change in vascular cells throughout. And so something about not just the diameter, but also the initial state of cells at day 0 or day 1, where we're missing these center populations, is driving some of these spatial constraints as well. And so, this got us really excited. We're going to be doing some spatial sequencing to understand more exactly what's happening over here at time. But one thing I really want to highlight is that, okay, well, we can do, say, immunofluorescence or these videos to kind of, semi-quantitatively understand what the cells are inside of our organoids. But what we really want to be able to do is pull these cells out from our organoids using different digestion protocols and actually be able to do like single cell immunofluorescence or single cell sequencing, calcium imaging, electrophysiological flow cytometry. And so we looked into the literature a little bit, just like Alice had done. And we know that most cardiac groups, is what we've done before, typically just use collagenase 4, as that's a key protein that cardiomyocytes generate and typically use inside of a cardiac tissue. What we had found from a semi-recent paper is that they had tested 3 different general enzymes, so acutase, papain, and triple E, which are more general proteases. And they had found, particularly for cardiomyocytes that have been derived from iPSCs, that papain, this enzyme that comes from papaya, seemed to have the highest yield of cells. And so we saw this and we were quite interested because we've had some challenges pulling the cells out of our organoids with high viability. And so this was done in conjunction with Dr. Giselle Gonzalez in our laboratory as well, a fantastic postdoc who I work with, where we were looking at sort of seeing if we can adopt this papain digestion for our lab and compare it to our standard collagenase 4 that we do. And so what we do is we take the papain and we add cofactor of some L-cysteine. We wash our spheroids with a little bit of EDTA to remove some of the calcium from around them. And then we sort of add the papain at different times and we do some cell counts. So we compared this to our collagenase 4 cocktail that we typically use, which also has acutase in it. So we can do matrix reduction with the collagenase 4 and then actually pull the cells apart from each other with the cell-cell junctions using acutase. And we would take our spheroids, in her case, and as well as our organoids that we've generated, and we put them into a little rocker inside of an incubator, and we would go for 10, 30, or 60 minutes. And so this is some really crude data, but this just gives you an idea of what we're sort of looking at when we're trying to give you the most real representation here. Is that with the collagenase 4s, you can kind of see when we're doing just generic cell counts, we get a lot of these chunky, clustery things that haven't really fully degraded properly. And we're just getting way less cells, and when we do, they're getting stuck in the filters, or we're getting a lot of doublets and triplets left. When we use papain, we actually got 2 to 3 times the yield of cells in the same amount of time. The collagenase didn't really seem to pull up more cells over time, so 10 minutes was already enough. Whereas a papain, was roughly a bit more strong at 60 minutes, but again, at 10 minutes, we're still getting a majority of the cells we want, so we can know that we're able to get a good yield very quickly. And so then we can use these cells that we've digested and actually sort of look at their functional physiology. And so here we're looking at calcium imaging of these monolayers of cells, and you might be able to tell here that for our papain cells, after an hour, they were able to reconnect with each other much stronger and actually be able to do much better calcium contractile, which is good, right? Because we know we can see these organoids and spheroids bouncing when they're balls, but then when we pull these cells apart, not only is it that we want to focus on getting live cells, but we want to make sure that they're functionally active as well. And that if we know if they're contractile before we digest, we got to make sure that they're contractile after they digest so that we can accurately compare our different organoids and spheroid structures and know that the digestion protocol is not interfering with that. So then this allowed us to actually do more downstream analyses. We were able to do individual cell fluorescence intensity as well, and sort of looking to sort of see that we actually had more of these cardiac cells in our smaller patterns, so alpha-actinin as well as CD31. However, this wasn't quite borne out when we had done our general immunofluorescence, and that's mostly because they're differently organized spatially, right? So the smallest patterns seem to have the most cardiomyocytes, but they weren't properly spatially organized, and so this made it a little bit harder to tell, as well as then we can do a lot more flow cytometry. And this is really nice because then this gives us really nice, good quantifiable data on the number of cells. And so again, we can sort of see as we increase our pattern size, we're getting a reduction in those cardiac cells. And this is really nice to pair where we can have this individual single cell flow data. We can directly compare to our 2D images of the whole organoid where we can actually see the full structure. And so with that, I just wanted to sort of summarize our project on the cardiac engineering side. Yes, we can generate cardiac organoids from micropattern hydrogels, and we can create these array of these organoids. And that digestion enzymes matter. So even in this context, we're not even taking a super complex human tissue with dozens of different ECMs that have been built up over the years, right? This is something that's only 7 days old, and already at the 7-day mark, we're having multiple types of ECM materials that are completely affected by the type of digestion media that we use in enzymes. And so we're still doing some more optimization and trying different protocols. I'm sure, as most of you guys know, these optimizations can go on forever. And so at some point you have to say, okay, we've got a good protocol and we're going to move forward. And we're happy with our pat pain setup that we've got. And as well as I've given you a little teaser on how pattern shape can influence not only the cell localization, so where these cell subtypes and cardiac cells are locating, but also the ratios of these different subtypes. And so we're exploring this further. We know that some of these cues are important. We're going to try and do some sequencing, spatial sequencing in single cell to understand what cues these cells are producing in these different spatial locations that drive these different patterning effects, and then see if we can use that to sort of create better and more representative organoid tissues that then we can begin to use our stem cells derived from patients at different ages and do different levels of maturation to begin to model sort of fibrosis or other types of cardiac diseases and pathologies that are age-related too. And so with that, we both want to acknowledge the Engler Lab, who has been super helpful in everything, particularly Professor Adam Engler, as well as Professor Sam Board, who have been super helpful with everything we've sort of done and given us the support we need for this, as well as our funding sources, the WU Side Performance Alliance, the NIH, as well as the Rita L. Atkins graduate fellowship that was given to Alice that can help support her work here too. And so with that, we're happy to take any questions. Hope you guys enjoyed. Thank you.
Thank you both, Tom and Alice. We have a lot of questions and we'll do our best to get to all of them. And so as far as jumping in, please, either of you feel free to answer the questions as I ask them. So starting off, our first comment slash question is super nice work. Any issues with the size of muscle cells and FACS sorting, especially for cardiac myocytes?
In my experience, size of the muscle, well, the bigger the muscle, I would say more cells I would get. But I think at one point, I was getting like really large samples from the tissue remnants of from ACL reconstruction surgeries, and there was time where I had like 4 grams of tissue, and that itself kind of presented more like issues in terms of like how fast I could mince the tissue. And at one point I was like digesting, or I guess mincing the tissue for about 45 minutes because I wanted to be thorough, and mincing the tissue really small would then, have implications downstream in terms of how well the digestion of the enzymes would work. So I think I was having like both of the problems where I would get really tiny samples and have low yields versus sometimes I would get really large muscle samples, and that would there would be like these time constrictions that I would try to do my digestion within a span of like 3 hours, but I would have to mince the tissue for like about 40 minutes. So I think that was one of the issues I was facing in
Thank
terms of
you,
the site.
Alice. So next question. Our lab has been using pronase instead of disbase. What's the advantages of disbase over other enzymes?
As far as I know, for this space, I was using it specifically to digest the extracellular matrix around satellite cells. So this space is a lot more specific in terms of digesting collagen 4, which is predominantly found in basement membranes. And it also digests fibronectin, which would, help with the isolation of both my satellite cells and also fibroadipogenic progenitors. If I'm familiar, pronase, I do believe that pronase is a lot more non-specific, and it digests all the, I guess, peptides and all the proteins. So in terms of like what you're trying to digest, I think it's important to kind of decide which protein would be better, for your application. And I think trying would be the best way to decide that.
Calcium imaging, what has been the ideal calcium concentration? Is it a stepwise addition, or do you bring it to the desired concentration at once?
Uh, yeah, calcium imaging is always a challenge, particularly when we're trying to do 3D or dissociated cells. Usually for every experiment, we do a titration. So even for each biorep, we're going to take usually like 0.5, 1, 2, or 4 micromolar. Fluorophore was the one we mainly use. Using reporter lines is honestly the best if you want to use calcium imaging. Obviously, you can't do that with human-derived or patient tissues, but if you're going to use stem cell-derived, that helps. And so every time we just make more samples and we do the titration. And sometimes you got to go a little higher for these cells that day, sometimes a little bit lower. And so we kind of just go with what seems to work best on that day. But we do it all in one addition. So we'll quickly add it and do like a sort of 30-minute incubation and then go. So we don't like slowly titrate and add it to the same cells. We'll just add to one set and then image them at their different concentrations.
And so the next question is we presume these are fresh human tissues that you used. What is the minimum quantity you used in milligrams? And did you try to optimize the protocol in mice or other preclinical muscle tissue?
So these were fresh human tissues, and the smallest quantity that I ever used was probably 100 milligrams, and I was able to get a good amount of yields to be able to do FACS sorting for satellite cells and FABs, even though the yield was a lot like lower than my other like larger tissues that I would get, but I was able to establish the cells that I wanted. And I did not optimize the protocol in mouse or other clinical muscle.
Thanks, Alice. And a follow-up question actually to that. And did the staining with your antibodies for flow separation affect cell behavior, survivability, viability, et cetera?
So for flow separation, let me see. Okay. So I did not necessarily test that. I would say I do include a viability for my flow cytometry with my flow cytometry antibodies. And I did notice that with the viability would be a lot higher for like younger patients, but viability would decrease with older patients. But I haven't noticed a change in viability in terms of the two different enzyme dissociations that I was using.
And so this is also a combination comment question. So our group has been using collagenase slash disbase for some time for satellite cell isolation as well as organoid digestion. As Alice mentioned, there have been a lack of good standards in reporting. One thing I found for collagenase and disbase, particularly collagenase, is the required cofactors, like calcium in this case, that are often not reported from most suppliers. Furthermore, each of these enzymes has alternative ways to stop their specific activity. And so the question is, do either of you have any suggestions or have found any standards for these required enzyme cofactors and neutralization methods?
So for my collagenase disbase combination, I do reconstitute my collagenase powders into HBSS that has magnesium and calcium, and those are able to provide the proper cofactors during my dissociation. So that was one thing that was actually mentioned in the Worthington data sheet for when I purchased the enzymes. And for when I do my digestion, I actually include or my digestion buffer itself is in 10% FBS. And one of the reasons I chose to do that is to provide the cells with some proteins and other like cytokines and from the FBS to help with their survival. And I had seen 10% FBS be used in other literature. And I do find that 10% FBS doesn't really affect the activity of the enzymes. But when I stop the digestion, I the way I neutralize the enzymes, I guess, at the end of the dissociation is by diluting it basically two times by more 10% FBS. So that way, and then following step is basically centrifugation, which then takes out takes the cells out of the digestion buffer.
Thank you, Alice. And just follow not on the topic of cofactors, just I wanted to provide a follow-up question, or what cofactors were used with acutase slash collagenase 4, if any, to digest the cardiac organoids? And Tom, if you want to take that one.
Yeah, I didn't actually have to use any cofactors for mine. I just put it into PBS without magnesium and calcium. And I think that is because my organoids are typically, I'm doing flow somewhere between 5 to 9 days old from stem cells. So they haven't produced a ton of matrix. I do know for people in our lab that have done, say, spheroid models that are 30 or 50 days old, that they've done a lot of more maturation with. Sometimes in those contexts, they have to explicitly add calcium or use calcium, magnesium, PBS for that. But for my younger tissues, they haven't produced as much matrix, or it hasn't really been a factor for me. So I think it really depends on sort of how much matrix your sort of conditions have. So say if you're working with spheroids that you formed 2 days ago, you probably aren't going to need them. But if you're working with something that, you've been culturing for a month or 2 or 3 months, so you're taking actual human patient tissues that are really tightly woven with really well organized matrix, then it's going to matter a lot.
Thanks, Tom, and this could probably be for either of you. That's comment slash question. Great work. Do you have any thoughts or experience on cold enzyme digestion or mechanical disassociation? These methods compared to warm enzymes might have an advantage to the study of metabolism and epigenetics.
It's a great point. I haven't actually worked with any of those before, other than, cold digestion in terms of like lysine, but then there's no cells left after that. So
Yeah, I don't also have experience with that.
Okay, I appreciate it. Do you have you observed any batch to batch effect among collagenase formulations in comparison to other enzymes? And relative to this, have you tried a defined collagenase formulation with known protease activity?
I will say that at least the ones that we've bought from Worthington, they're pretty consistent. But I do know brand to brand is much more significant in terms of enzyme activity. So some people will say 50 units for one brand. It doesn't really equal the same digestion capacity as 50 units from another brand. Usually when you stay within the same brand, it's not that bad. I don't know, Alice, if you've had any differences with yours over the years.
I would say it has been pretty consistent. The one thing I would mention though, that I think each time you buy a new batch of enzyme, they come with their own specific units per like gram of enzyme. So then you would have to when you're reconstituting or using, you'd have to take that into consideration. So each batch may not be the same volume that you need to use for your digestion. So gotta kind of have to take that into consideration.
Both. And now the next question, which likely may be our last question, given that we are close to our allotted time, and please keep in mind if this is too specific, then I can perhaps provide you the attendees' contact information for follow-up. So the comment question is, I just started studying immune cells in the muscle, but I keep getting different data even in saline controls, mice muscles. How can I reduce variability in flow cytometry data? Could it be due to inconsistent mincing? Enzyme composition in the digestion kit. She notes the kit does not specify the enzymes used or incubation conditions.
I could comment on that. So in terms of variability, I would definitely agree that inconsistent mincing could provide some variability. In my digestions, I've definitely noticed if I don't mince the tissue finely enough, my digestion doesn't really go efficiently. And at the end of the digestion, I would notice like still muscle chunks left over even after 90 minutes of digestion. So I think mincing consistently and small, really small chunks and to basically create a slurry of the muscle would potentially increase the consistency of your results. In terms of the oh, I lost the question. I think for enzymes, maybe just make sure that for the amount that you're using is consistent per gram of tissue. So don't just use, let's say, 10 mLs of the digestion buffer for anywhere from like 1 gram to 2 grams of muscle. So scale up the volume of the digestion buffer you're using if your muscles are larger, let's say, from one experiment to another.
And a practical aspect, I mean, I guess if this is something you're exploring, I mean, honestly, I would just suggest maybe split one of your muscles in half, mince the heck out of one of them and leave another one like kind of minced. Because especially with immune cells and sort of muscles from animal models, like it could be so variable animal to animal too, and muscle to muscle, that it's going to be really hard to tell if it is your protocol or just biological variability, right? So you'd probably really want to take the same muscle, split it in half, maybe in thirds, depending on the cell yield or the yield of your muscle, and sort of test some of those on the same muscle, just to be sure that either it's all reproduced within that same muscle, and then you know sort of exactly what's causing that problem. So you can also try different concentrations of the enzyme kit too, and sort of see if I have the same muscle split in half, maybe that's why I'm getting different enzyme amounts or immune cell amounts.
Thank you both. And I think that'll conclude our Q&A portion of today's event. I just want to thank you both for providing us with your time and insights and very thoughtful responses to our many Q&A questions and submissions. We want to thank Worthington Biochemical for supporting APS's mission, as always, to provide valuable education, and everyone in our audience today for taking the time to join us. Before you sign off, we hope that you'll take a few minutes to complete a short survey about today's session and suggest new topics for us to cover in future events. Thank you very much. Have a good day, everyone.
Thank you. Bye.
Take care.